Hello everyone. Today lecture is about the transport across the cell membrane, including osmosis. Sit back, relax and enjoy our presentation. But we'll start by an introductory part regarding body fluids. Water constitutes about 60% of our body weight. This percentage is less in the obese persons and in the females. This is because the fatty tissues contain less water. However, this percentage is more in infants. The 60% of our body weight water can be divided into two compartments: the intracellular fluid compartment and the extracellular fluid compartment. 40% of the total body weight is intracellular fluid, divided between body cells and blood cells. The extracellular fluid compartment, the 20% constitutes 20% of our body weight, is divided between the plasma around the blood cells, 5%, and the fluid outside the body cells, what do we call the interstitial fluid, 15%. The ratio of interstitial fluid to the blood plasma is 3:1. This is because the interstitial fluid acts as a reserve for the blood in case we lose blood in hemorrhage. Even before blood transfusion, a good amount of water will pass to the blood. First, we'll compare the intracellular fluid with the extracellular fluid in a table form of 5 points. I like to be very organized. They differ in 4 points, but they share only 1 point. The first difference is regarding the volume. As we already said, the intracellular fluid compartment constitutes 40% of the body weight, while the extracellular fluid compartment 20%. In multiple choice question, you may have the ratio. You will say that the intracellular fluid is double the extracellular fluid volume. Correct statement. Or the ratio of intracellular fluid to extracellular fluid is 2:1. Again, it's a correct statement. The second difference is regarding the chief cations. That's the positively charged ions. They are called cations as they migrate to the cathode in the electric field. The chief cation in the intracellular fluid is potassium, while the chief cation in the extracellular fluid is sodium. This does not mean that potassium exists only intracellular and sodium only extracellular in the extracellular fluid. This is wrong. Potassium and sodium, both of them exist inside and outside the cells. But potassium is the chief cation intracellular, while the sodium is the chief cation extracellular. The symbol for potassium is the letter K. This is an Arabic expression because potassium is responsible for the alkalinity of the body fluids. In Arabic language, we call it qalawi. They wrote it in English instead of qalawi, there is no qaf, they wrote it kalium. This is why the abbreviation, the symbol for potassium is not P but K. Again, the symbol for sodium is Na. This is an Arabic word also, because it's related to the valley of Natron in Egypt. Natron, this is where the sodium was found, so they call it Na. Now the third difference is regarding the chief anions. The chief anions in intracellular fluid are the proteins and the phosphate, while the chief anions in the extracellular fluids are the chloride and the bicarbonate. I have a very simple way for you to memorize it in a very easy way. The main ions inside the cells all sharing the letter P: potassium, proteins, and phosphate ions, while the main ions outside in the extracellular fluid, they have no P letter. Sodium is the positive ion, chloride and bicarbonate are the negative ions. The fourth difference regarding the pH. The pH is an abbreviation of the power of hydrogen, or in Germany, Potentialhydrogene. This is the origin of the abbreviation pH. Think with me. Do you think which is more acidic, the intracellular or the extracellular fluid? You have to know that our body metabolism does not produce except acids. These acids are produced inside the cell because the metabolism, the metabolic processes occur inside the cells. So the intracellular fluid is more acidic than the extracellular fluid. Acids are produced inside the cells and then migrate to the extracellular fluid where they are buffered. So the extracellular fluid is less acidic. So the intra and the extracellular fluid differ from each other in 4 points. The volume, the intra is double the volume of the extra. The second point, the chief cations and chief anions for intracellular fluid, all sharing the letter P: potassium, proteins, and phosphate, while for the extracellular fluid, sodium, chloride, and bicarbonate. The fourth difference is the pH, the power of hydrogen. The intracellular fluid is more acidic, the extracellular fluid is less acidic. However, the intracellular fluid and the extracellular fluid share only one common property, that they have the same osmolality, which is a very important number to be remembered. 290 milliosmoles, not osmol. This is because the cell membrane is freely permeable to water. This results in equal osmolality on both sides because the cell membrane allows the passage of water in and out from the cell until both will have the same osmolality. That's 290, open your ears, milliosmoles, not osmoles. Or we can say it in a simple way, 300 milliosmoles. This table form with its 5 facts is very, very important in multiple choice questions. What numbers you have to remember about the sodium, potassium, chloride, bicarbonate, and so on? In the medical labs, we analyze the extracellular fluid. The samples are taken from the extracellular, not from inside the cells. So the important numbers are those related to the extracellular fluids. You have to know that the sodium in the extracellular fluid in the blood or serum, very important number, 145 milliequivalent per liter. It combines mostly with chloride and bicarbonate. The chloride, 115 milliequivalent per liter, and the bicarbonate, 28 milliequivalent per liter. You have to notice the followings. Number one, as most of sodium is united with either chloride or bicarbonate, the sum of the two numbers for chloride and bicarbonate almost equals the number for the sodium. 115 plus 28, the sum will be 143, almost as sodium, because most of sodium is combined to either chloride or bicarbonate. The very small difference is very important. We'll discuss it with the acid-base balance. It's called the anionic gap. There is a small difference because not all of the sodium is united with chloride or bicarbonate. The difference is we call it the anionic gap. It's very important in acid-base balance. The second thing I would like you to notice, if you combine these numbers, it will be 288. It's very close to the osmolality of the blood, because the sodium combined with the chloride and bicarbonate, these ions are the main ions responsible for the osmolality of the blood. This is the second point to be noticed. Even I would like you to know a fourth number. That's the potassium in the extracellular fluid, because as I already said, we measured the concentration of ions in the extracellular fluid. The potassium in the extracellular fluid, the normal potassium in the extracellular fluid, remember that, open your ears, should be less than 5 milliequivalent per liter. It should be 4.5 milliequivalent. However, it should be less than 5 milliequivalent. Now we come to expression called homeostasis. We have two similar expressions, and they are trying to confuse you in the multiple choice questions. Hemostasis and homeostasis. The word heme means iron. Most of iron exists in our blood. 70% of the iron exists in our blood. This is why the word heme may mean not iron but blood. Hemostasis, stasis means to keep constant. Hemostasis means stoppage of bleeding. While homeostasis, home is the place where you live. To keep the place where you live constant. I will say it in a very easy way. The fish lives in water. The body cells live in water. That's the extracellular fluid. This is the home of the fish, and this is the home of the cell. For homeostasis means to keep the internal environment, mainly the extracellular fluid, constant. You have to complete the statement. You have to keep it constant for optimum function of cells. You cannot say that homeostasis is just to keep the internal environment, that's the extracellular fluid, that's the home of the cells, constant, and you stop. You should keep it constant for optimum function of cells. The temperature should be kept at 37 degrees centigrade. The pH should be kept at 7.4, which is alkaline. Did you think ever why the pH of our body is alkaline, 7.4? Think about it. I already said that the body metabolism produces only acids. So we need our body fluids to be alkaline, 7.4, to neutralize these acids. What is the definition of homeostasis? To keep the internal environment That's the extracellular fluid constant for optimum function of cells. In multiple choice questions, note these facts. Homeostasis is maintained by most systems, not by few systems. Most systems of your body share in the process of homeostasis. What is the importance of homeostasis? The third point. Homeostasis is essential for life. That is, for living, growing, and for the functions of most organs. It ensures constant physical and chemical internal environment despite changes in the external environment or inside the body, outside the body or inside the body, despite external or internal changes. The fourth point, homeostasis, includes two changes. The changes between the intracellular fluid and the extracellular fluid, here we mean the interstitial fluid, and the second change between the interstitial fluid and the blood. So the homeostasis depends on two changes between the intra and interstitial fluid, intracellular fluid and interstitial fluid, and interstitial fluid and the blood. The fifth point, to give an example, we have in homeostasis to keep the extracellular fluid constant regarding the temperature, regarding the pH, regarding the volume, regarding the osmolality, regarding the nutrients, regarding the ions. We take the temperature and pH as examples. To keep constant temperature, you have to keep the heat production exactly equals the heat loss. To keep the pH constant, we buffer the acids produced by metabolism via chemical buffers. But this is not sufficient, because chemical buffers, all what they do, they can change strong acids into weak acids. Still, we have to get rid of the acids. This is the process of the lungs and kidney. Inside the body, we have two types of acids. We have volatile acid, that's the carbonic acid. Because by the aid of carbonic anhydrase enzyme, carbonic acid is broken into carbon dioxide. This we can get rid of through the lungs. So the lungs are responsible to get rid of the volatile acids. We mean the carbonic acid, which is broken by carbonic anhydrase enzyme into carbon dioxide and water. Water, no harm, but we get rid of the carbon dioxide. The other acids, we call it fixed acids, like sulfuric acid, phosphoric acid, lactic acid. These acids we have to get rid through the kidneys. So the lungs, the kidneys, the buffers share to keeping the pH. To keep the temperature constant, we have to make sure that the heat production equals the heat loss. Now we start the main core of our lecture. That's the transport across the cell membrane. We have three ways of transport across the cell membrane. Number one, diffusion, which is a passive process. Number two, the active transport, which as the name indicates, is an active process. And number three, the vesicular transport, which is also an active process. So very important MCQ fact. Diffusion, by all its three types or subtypes, is the only passive transport across the cell membrane. Diffusion is the only passive way of transport across the cell membrane. The other two ways, the active transport and the vesicular transport, both are active process. Now we start with diffusion. Every information, every even bit of information is a multiple choice question fact. I like to be very organized. we'll discuss diffusion in 4 points. Number 1, diffusion occurs with gradient or along gradient or down gradient. You may face one of these 3 expressions in the multiple choice questions. Diffusion occurs with, along, or down gradients. I always say it's as if a child is going down the slide. This child does not need any energy to go down. The same in case of diffusion. So the second point in diffusion, you can expect it from the first point. As diffusion occurs with or along or down gradient, so it's passive. And I said it is the only method of transport across the cell membrane which is passive. That means it does not need adenosine triphosphate, which is the source of energy in our body. One may ask me, what do you mean by gradient? For solutes, they diffuse down concentration gradient. For gases, down pressure gradient. For ions, when positive ion attracts negative ion or the reverse, this is electrical gradient. So we have 3 types of gradients. For solute, concentration. For pressure, for gases, pressure. For ions, electrical. Sometimes the ions diffuse along both concentration gradient from higher concentration to lower concentration, and again with electrical gradient. This means that positive ions attract negative ions. In this case, we do not say that ions diffuse along concentration electrical gradient. We call it electrochemical gradient. When ions diffuse along both electrical and concentration gradients, we call it electrochemical. So we finished with 2 parts, with 2 points in diffusion. Diffusion occurs with, along, or down gradient. And now we know the types of gradients. And being with or along or down gradient, it's a passive process. It does not need ATP. And I remind you for the 3rd time, diffusion is the only transport which is passive. The other 2 are active. Now we come to the factors affecting diffusion. Actually, you have a lot of factors affecting diffusion, but for now we will discuss only 5 of them. There are not only 5s, we will discuss 5. Factors affecting diffusion. Diffusion is directly proportional to concentration gradient. The more the concentration, the difference between C1 and C2, the more will be the rate of diffusion. So diffusion is directly proportional to concentration gradient. Definitely, diffusion is directly proportional to the area. The diffusion of gases in both lungs is better than the diffusion in one lung. Why? Because 2 lungs have more area than 1 lung. Diffusion is directly proportional to the temperature, because the temperature, the heat gives the molecules the energy to move. So diffusion is directly proportional to the temperature. And here we have a very important multiple choice question fact. At absolute zero temperature, the diffusion rate will be 0. There will be no diffusion. Now we come to the diffusion is inversely proportional to molecular size, because the higher the molecular weight, the larger the size of the molecule, definitely the less will be the rate of diffusion. So diffusion is inversely proportional to, but not to the molecular weight, but to the square root molecular weight. This is very important point. Try to the square root. Try to understand with me. We can imagine the molecule as a circle. The size of the size of the circle, the size of the circle depends on their radius. Radius power 2. The square area of the circle, the area of the circle, we can consider the molecule as a circle. The area is directly proportional to the radius power 2. So if the diffusion rate was directly proportional to the molecular size, we will say that diffusion rate is directly proportional to the molecular weight power 2. But it's inversely proportional to the molecular size. Larger molecules diffuse with difficulty. Smaller molecules diffuse with easiness. Then, what is the reverse of power 2? It is the square root. This is why we say that the diffusion rate is inversely proportional to square root molecular size. And I give an example which is an MCQ fact. What is the ratio? We have substance A, the molecular weight, 4 times that of substance B. Definitely B will diffuse more. But what is the ratio? We take square root of 4:2, and then you switch B, which is smaller, will diffuse 2 times more than A. So to answer this question, you have to do 2 things. First, to reverse, because the bigger will diffuse less and the smaller will diffuse more. Then to take the square root. The square root of 4 is 2. And now we know why we take the square root. Because if it was directly proportional, the size of the molecule increased directly proportional to the area of the circle, which is related to radius power 2. Being inversely proportional, the reverse of power 2 is the square root. This is why we take the square root. Now we come to the second factor. Diffusion is inversely proportional to the length of diffusion. That's the thickness of the membrane. That's the distance of diffusion. Let's summarize all these factors in one equation. The diffusion rate is directly proportional to, we said, 3 factors. Concentration gradient, that's C1 minus C2, area of diffusion, number 2, and the temperature. And is inversely proportional to the square root molecular weight and the length of diffusion, that's the distance of diffusion, that's the thickness of the membrane. So we finished 3 points in diffusion. It occurred with, along, or down gradient. So it's a passive process, it needs no ATP. And we discussed 5 factors. Diffusion rate is directly proportional to concentration gradient, area and temperature, and inversely proportional to square root molecular weight and length of diffusion. Now we come to the more important, the last point, which is the type of diffusion. Types of diffusion are 3. Simple diffusion, facilitated diffusion or carrier-mediated diffusion, and the third type is osmosis. So we'll discuss the 3 types: simple diffusion, facilitated diffusion, which what do we call carrier-mediated diffusion, and osmosis. We start with simple diffusion. In order to understand physiology, I want you to use 2 of the characters you have. Being thinker, you can think with me, and imaginative. And by the way, thinking and imagination are not related only to human beings, but to other higher animals as well. For example, chimpanzee and orangutan. So try to think with me and to imagine with me. This will make the subject very easy. We start with simple diffusion. Imagine the wall in front of you, or the page in front of you, is the cell membrane. This cell membrane is nothing but a lipid substance. Floating in this lipid substance, protein channels. That's imagined as a protein balls. Each ball contains a channel, and the protein channels in turn contain water. So I want you to imagine the page in front of you is the cell membrane. It's nothing but lipid substance. Floating in this lipid substance are protein channels. These protein channels contain water. But very important point to note that the area of the protein channels 1/100 the area of the membrane, because we already said that the diffusion rate is directly proportional to the area. Then I will ask you a question. Consider yourself the substance. You have two ways to cross the cell membrane. Either to cross via lipid substance or to cross via the protein channels. Then you expect how many ways the simple diffusion has. One may answer two ways. You already said we have a lipid substance and protein channels. The substances may pass through the lipid substance, or the molecules may pass through the lipid substance or through the protein channel. Actually, the correct answer is simple division has three ways to occur. For the molecules, they can pass through the lipid substance, number one, protein channels, number two, or both protein channels and lipid substance, number three. Think with me. What do you think the character of the substance should be in order to pass through the lipid substance? I think they should be lipid soluble. For lipid soluble substances or molecules, they can pass through the lipid substance. The membrane is lipid and they are lipid soluble. There is no problem. So they will pass through the lipid substance. And I tell you something, this is the best opportunity. You know why? Because the area of the lipid is 100 times the area of protein channels. And definitely the diffusion rate will be directly proportional to their lipid solubility. And here, 6 factors affecting the diffusion rate we did not mention in the page before. For molecules which are lipid soluble, the more the lipid solubility, the more will be the rate of their diffusion. And you know what is the best example of this? Is oxygen. And I have thing to say over here. You know, all your body cells, what is the most important element for all your body cells? Your body cells cannot withstand deprivation from this element for seconds. That's oxygen. This is why oxygen is highly lipid soluble. I tell you something, oxygen crosses the cell membrane as if the cell membrane does not exist, because it's highly lipid soluble. Don't ever forget this example. So for molecules which cross the cell membrane by simple diffusion, they can cross through the lipid substance if they are lipid soluble. And the more their solubility in lipid, the more will be the rate of diffusion. And the best example is oxygen. Now we come to the second possibility. Imagine if molecules are lipid insoluble, so they missed their chance to pass through the lipid substance. to think for another way. They can pass through the protein channels which contain water. So there must be two additional characters. To pass through the small protein channels, they have to be of small molecular weight. And these protein channels contain water, so they have to be water soluble. Now, you can know or expect what are the characters of the molecules to pass through the protein channels. Why they go to the protein channels and miss their higher chance for lipid substance to pass through the lipid substance? Because they are lipid insoluble. They are now forced to pass through the protein channels. Protein channels constitute only 1/100. They are very small of the area, so there must be of low or small molecular weight. Protein channels contain water, so there must be water soluble. And the best example for here, the best example is iron. All ions pass through the protein channels. What governs the passage of ions across the protein channels? Two things. First, selective permeability. Why protein channels will select one type of ions and not the other type? This is what do we call selective permeability. This depends on three factors. Let's think. Definitely the most important factor is the diameter, the relative diameter of the ion in relation to the diameter of the protein channels. If the ions are larger than the protein channels diameter, definitely you will not pass. The second thing, their shape. You know what? Ions inside our body, all of them are hydrated ions. Somebody may ask me, what's meant by hydrated ions? They are covered by a shell of water. And this explains why in the nerve the permeability for the potassium is 20 to 100 times the permeability to sodium, despite the fact that potassium ion is larger than sodium ion. You know why? Because hydrated potassium in the body is smaller than hydrated sodium. This explains how a larger ion regarding its molecular weight like potassium diffuses much more easy in the protein channel than sodium, which is the smaller ion, because sodium collects a large amount of water around the ions, so it cannot pass as easy as potassium. The hydrated potassium ions are smaller than the hydrated sodium ions. Thirdly, it self-explains the charges. If we have positive ions and the channels have negative ions, they will attract each other. But if they are of the same type, positive and positive, repulsion will occur. So the selective permeability depends on three things. The most important, the diameter. Second thing is the shape. The third thing is the charges. Now we come to the gating. Some channels have no gates. We call it non-gated. So they are open all through, all the time. Other channels are gated. We have two types of gates. I always say that physiology is like life. In life, we have two types of gates. Electric gates, that is, gates which open by electrical changes. We have this type in the body. What do we call voltage gated channels? And we have another type of gates called ligand gated, like the door of your house. It's not an electric door. It's opened by a key, which is the ligand. But you do not put the key in any place in the door. You have to put it in a special location, which is the lock. In the cell, this special location is the receptor. The union of a ligand with the receptor will open the gate. We call this gate ligand gate. And the ligand here is the key. In the case of your home door, in the body, it is a chemical substance. And like physiology, like life, exactly, we can open the door from inside or from outside. So we have external ligand like hormones, for example hormone, and we have internal ligand like calcium and cyclic AMP. But I would like to remind you that simple diffusion for molecules that diffuse through the protein channels, the diffusion rate is much less than molecules that diffuse through the lipid substance. You know why? Because the area of the protein channels is 1/100 the area of the lipid substance. So the best opportunity, the best chance is given to the molecules using the lipid substance, the molecules which are lipid soluble, and the best example is oxygen. Less chance for molecules which are lipid insoluble. They have to use the protein channels. They have to be small molecular weight because the channel is small, and they have to be water soluble because the channel is filled with water. And the best example here are ions. And this depends on selective permeability and gating. The selective permeability depends on diameter, shape, and charges. The gating, we have two types of gates: non-gated channels and gated channels. We have two types of channels: non-gated channels and gated channels. Gated channels are of two types, exactly like life. Voltage gated, that's gates open by electrical changes, and ligand gated, that's gates open by a ligand. This ligand may be external ligand like hormones, or internal ligands, for example calcium and cyclic AMP. Now we come to the third way of simple diffusion, is to pass both in the lipid substance and protein channels. I always say, which substances, what are the examples of the substances that can pass in both the two, the lipid substance and the protein channels? We have two examples. So 2 for 2. We have water and water soluble substance. Water and water soluble substance, for example carbon dioxide and urea. These substances must have two characters. They must be of small molecular weight, low molecular weight, to be able to pass through the protein channel. And they must be uncharged to be able to pass through the lipid substance despite the fact they are lipid insoluble. The water is lipid insoluble. The water does not dissolve in oil or lipid. How water molecules can cross this lipid substance? They can cross this lipid substance as bullets, depending on the very high kinetic energy. So these molecules must be uncharged. Otherwise, if they are charged, they may face in the cell membrane some charges of their type to cause repulsion, to cause decrease in their kinetic energy. Then they will fail to pass through the lipid substance despite the fact they are lipid insoluble. They cross the lipid substance despite they are lipid insoluble because water does not dissolve in lipid and internal water soluble substance. They cross the lipid substance depending on their high kinetic energy as bullets. So they must be uncharged. And the example here is water and water soluble substance, for example carbon dioxide. To summarize, in simple diffusion means that we need no help. We do not need any facilitator. This is what's meant by simple diffusion. The cell membrane, as I already told you, consists of lipid substance with protein channels floating in this lipid substance. Then we have three ways to cross the cell membrane. To pass through the lipid substance for lipid soluble molecules. If the molecules are lipid insoluble, their only chance is to pass through the protein channel. They must be small molecular weight and water soluble because the channel contains water. And there are some molecules pass through both. The two ways, we have two examples. For the two ways, we have two examples. Water and water soluble substances like the carbon dioxide and urea. And they must have two characters, being small molecular weight to be able to pass through the protein channels, and uncharged. Depending on their high kinetic energy, they will cross the lipid substance despite the fact that they are lipid insoluble as bullets. Now we come to the second type of diffusion. We finished with the simple diffusion. We come to the second type. Before I started with the second type, let me ask you a question. Glucose. Do you believe that glucose is an important substance? Definitely yes. Glucose is very important substance. It's a very important source of energy for most of cells. Then let me tell you the specification of the glucose molecules and try to think with me. Glucose molecules are lipid insoluble. Then one may say then glucose molecules missed their chance to pass through the lipid substance because they are lipid insoluble. Then we think about the protein channels. But to pass through the small protein channels, the molecule should be small, of low molecular weight. Glucose molecules have high molecular weight, 180. And now glucose molecules missed both chances to pass through the lipid substance, being lipid insoluble, and to pass through the protein channels, being of high molecular weight. However, glucose molecules are water soluble. Let us think how to solve this problem. We have to help glucose molecule. We have to facilitate glucose molecules. This is why it's called facilitated diffusion. This is done through a carrier. This is why facilitated diffusion is also called carrier-mediated diffusion. We will discuss it in 4 points. We already finished with the example, one of the points. The best example is glucose. Why we have to facilitate glucose? Because glucose molecules cannot pass through the lipid substance, being lipid insoluble, and cannot pass through the protein channels, being of high molecular weight. But fortunately enough, they are water soluble. Now we come to the mechanism. The only thing is that we have to widen the protein channels for the large glucose molecules to pass through. I tell you something, physiology, like life. In life, if you drill a hole in the wall, usually the outer part is wide and the inner part is narrow. The same in the cell membrane. The outer part is wide and the inner part is narrow. Now glucose binds to receptor on a carrier protein in the wall. Glucose molecules bind to receptors on the carrier protein by aid of an enzyme. This will lead to configurational change change in the shape of the protein channels, they become wider. The inner part will get wider, so glucose molecules can pass through. So it's very easy. We facilitate the diffusion of glucose using a carrier and enzyme. This is why it's called facilitated diffusion or carrier-mediated diffusion. The glucose molecules bind to receptors on the carrier protein by the aid of an enzyme. This will lead to widening of the inner part of the channels so that glucose molecules can pass through. Now we come to the fourth point in facilitated diffusion. What are the differences between facilitated diffusion and simple diffusion? We may call it characters of facilitated diffusion or differences between facilitated diffusion and simple diffusion. Depending on your thinking. Think with me. What's new in facilitated diffusion? Smart student said, two things are new. We got an enzyme and we got a carrier protein. So the differences for a smart student must be related to either the enzyme or to the carrier protein. The sharing of an enzyme in the process. Enzymes are known to be highly sensitive to temperature changes. So in facilitated diffusion, which depends on the presence of an enzyme, will be in turn very sensitive to temperature changes compared with the simple diffusion. The facilitated diffusion are 10 times more sensitive to temperature changes than simple diffusion. We know that simple diffusion also is sensitive to temperature. We took temperature as one of the factors affecting the simple diffusion. But in facilitated diffusion, because of the presence of the enzyme, facilitated diffusion is very sensitive, highly sensitive, 10 times sensitive to temperature changes. Then we come to the second thing, which is the carrier protein. This carrier protein has many receptor binding sites for the molecules to bind with. I explain it in a very easy way. Let's imagine this is the carrier protein, and these are the substances, the molecules trying to utilize this carrier protein to enter the cells. We have molecules A, molecules B, molecule C, molecule D. Look to the diagram. In your opinion, which molecules of these four molecules can use this carrier protein? One may say A molecules. I said wrong. A or B? It depends on the structure, not on the color. The structure is the most important factor deciding the molecules will fit or will not fit inside the receptor binding sites. For both A and B can fit in this configuration, in this shape. C and D cannot. So not all substances can utilize the carrier, only the substance with structural specificity. So the structural specificity is one character in facilitated diffusion. Not any molecule which is lipid insoluble, large molecular weight, water soluble, can diffuse via facilitated diffusion. Its structure, the structure of this molecule should be specific to the carrier. So we have structural specificity. The second character related to carrier protein. Imagine A and B molecules. If the number of A molecules are increased, so they will occupy more receptor sites. Less receptor sites are left for B. So the second character will be competition. The higher the concentration of one substance utilizing the same carrier, the more its chance to diffuse than the substance with less concentration. Now we come to the third and fourth character. Imagine we have A molecules only. With increased concentration or number of A molecules, more receptor sites are occupied and more molecules diffuse. More increase in A, the number of A molecules, more receptors are engaged until by the end, all the receptors are engaged with molecules, no more receptor for new molecules, then we reached, the diffusion will reach a saturation or maximum. So the presence of a carrier protein leads to three characters. Structural specificity, only molecules with certain structure can utilize this carrier protein. A competition between substances use the same carrier protein, and even if there is one substance utilizing the carrier protein, the diffusion rate will reach a maximum when all sites are saturated. Saturation will occur. And here we have a multiple choice question curve. They will give you two curves, the one in black and the one in red. The diffusion rate is directly proportional to concentration gradient. In case of diffusion, simple diffusion, there is no saturation because there is no carrier. The more the concentration, the more the molecules will diffuse. But in case of facilitated diffusion, the diffusion rate will reach a maximum when all receptor binding sites are saturated. We reach a maximum. This is the facilitated diffusion. This was the second type of diffusion, which is facilitated diffusion or carrier mediated diffusion. And don't ever forget, being a type of diffusion, a very important MCQ fact. It's still passive, which means it does not mean ATP. The diffusion, the three types of diffusion, all of them are passive. They do not need ATP. Now we come to the third way of diffusion, which is osmosis. Osmosis is a very important subject in medicine because it's related to solution therapy, giving your patient solutions. You must know everything about osmosis, because if you give the patient hyperosmotic or what do we call hypertonic solution, it will drag water out of the cells will shrink. The reverse, if you give a fluid hypoosmotic or hypotonic in regard to the plasma, fluid will enter the cells will swollen and rupture. So we have to know everything about osmosis. It's a very important subject in medicine. Let's start with osmosis. I like to be organized as usual. I have 7 points to discuss in osmosis. Number 1. Before I start the definition, look to this U-shaped tube. Here we have a semi-permeable membrane, a membrane that allows only water molecules to pass through, but not salts or molecules of solute. In one limb we have pure water, only water. no solute. In the other limb, we have water and solute. What's diffusion? What's osmosis? Osmosis is a type of simple diffusion. It's a simple diffusion of water, not solute, because here the membrane is impermeable to solutes. It's only permeable to water. And we apply the same principle. Water will diffuse from the limb where the concentration of water is higher to the limb where the concentration of water is less. So water will diffuse in this direction, from the limb where we have more water to the limb where we have less water. They can confuse you in multiple choice questions. The limb which has more water definitely has less solute. The limb which has less water have more solute. So they may say to you, and it's correct statement, water will diffuse from the limb with less solute, because this limb has more water, to the limb of more solute, because this limb have less water. They may give you the statement in term of water or in term of solute. But remember that in osmosis we are talking about water, not solutes. From the limb where we have more water, that's we have less solute, to the limb where we have less water, that's we have more solute. Now we come to the first point of the 7 points of osmosis. Definition. Osmosis is a simple diffusion of water. The second point, they try to measure the osmosis force. It's difficult, but we have an idea. If we measure the force which stops osmosis, this definitely will be equal to the osmosis force. So it's measured by an intelligent or smart way. By what do we know as osmotic pressure? It's the pressure needed to stop osmosis. Definitely it will equal to osmosis. So they measure the osmosis force by measuring the pressure needed to stop this osmotic force. What do we call osmotic pressure? It is the pressure needed to stop osmosis, and it's measured in millimeter mercury. Now we come to the third point in osmosis. The osmotic pressure depends, choose the correct answer, on the number or the size of molecules, or both the number and the size of molecules. The correct answer, the osmotic pressure depends only on the number and not the size of the molecules. One may ask me, please explain it to us. We all know about the kinetic energy of the molecules. Why it depends on the number and not on the size of the molecules? The kinetic energy of a molecule equals half mass V square. So it's related to the mass of the molecule and the velocity of the molecule. It's very simple. The large molecule, because it is large size, its velocity is small. The reverse, when the molecule is small in size, because it's small in size, the velocity is high. So one thing compensates for the other. High molecular size, less velocity. Low molecular size, high velocity. Eventually, the kinetic energy for both molecules will be the same. So what matters is not the size of the molecule, because both they have the same kinetic energy. What matters is the number of molecules. So the osmotic pressure depends on the number, not the size of molecules. We come to the fourth point. What's mole? The word mole can be written in two ways. In 4 letters, M-O-L-E, or 3 letters, M-O-L. This word is derived from the first 3 or 4 letters of molecular weight. The mole is the molecular weight expressed in grams. And definitely the millimole is the molecular weight expressed in milligrams. And this is more important. You know why? Because most of the substances in our blood exist in milligrams, not in grams. In blood, with the exception of hemoglobin and plasma proteins, most of substances in our blood exist in milligrams. So the more important is the millimole. But for simplicity, I will explain by saying the mole, just to abbreviate the term. But instead of saying millimole every time, I said mole. Then what's mole? It's driven from the word molecular weight. The mole is the molecular weight expressed in grams, and the millimole is the molecular weight expressed in milligrams. What's osmole? The word expresses itself. Osmole is the osmolality caused by a mole. The osmolality caused by a mole. One may ask me, please explain. We know before that if every molecular weight, if we have the molecular weight of any substance in grams, it will contain the same number of molecules. What do we call Avogadro's constant? Avogadro's constant, which is, I think, if I can still remember, 10 power 23. Let's say, if you have a mole of glucose, or galactose, or fructose, or lactose, a mole, that's molecular weight of any of these substances, this mole and this mole and this mole, all of them will contain the same number of molecules. The same number of molecules. And you said that the osmotic pressure depends on the number of molecules. All of them contain the same number. So all of them will give the same osmolarity, as osmolarity depends on the number, and all of them have the same number. This is what we call osmole. That is the osmolality caused by a mole, because the mole from any substance contains fixed number of molecules. Avogadro's constant, we know. Smart students say to me, this is applicable to the non-ionizable salts. You mentioned in your example glucose, galactose, fructose. But if we are dealing with ionizable solute, we cannot apply this like sodium chloride or calcium chloride. I say why? He said to me, if we brought a mole of sodium chloride, theoretically, and we calculate the number of dry molecules of sodium chloride, it will be the same number as glucose or galactose, the same number. But when we put this mole of sodium chloride inside water, what we calculated as one molecule now will be ionized into 2 ions in case of sodium chloride. And you said that the osmolality depends on the number. Now the number of particles will be double. Each molecule of sodium chloride, dry sodium chloride, now changed into 2 ions, 2 particles. So 1 mole of sodium chloride, 1 mole of sodium chloride will give 2 osmoles. Yeah. Then to have 1 osmole of sodium chloride, 1 mole gives 2 osmoles. I want just 1 osmole of sodium chloride. I have to take half mole. That is the molecular weight divided by 2. I'll give another example. 1 mole of calcium chloride, CaCl2, calcium chloride 2. if we get a mole of dry calcium chloride, 1 mole will contain the same number. But when you put calcium chloride in water, what you calculate as 1 particle of calcium chloride now is changed into 3 ions, calcium and 2 chlor ions. And you said that it depends on the number. So the mole of calcium chloride will give us 3 osmoles. So to get 1 osmole of calcium chloride, you will not have to have a mole. A mole will give you 3 osmoles. You have to have 1/3 of this mole. Let's put a rule for that. So the osmole will equal the molecular weight divided by the number of ions of molecules. In case of sodium chloride, to get osmole, 1 mole of sodium chloride gives you 2 osmoles. So to have 1 osmole divided by 2. 1 mole of calcium chloride gives you 3 osmoles. So to have 1 osmole of calcium chloride divided by 3. And you know, this equation holds good even for the non-ionizable substance as glucose, galactose, fructose. You know why? Because in this case, you will divide the molecular weight by 1 because there is no ionization. So this is very important equation. The osmolality equals the molecular weight divided by the number of ions per molecule. Molecular weight divided by the number of ions per molecule. Now we come to the 6th point. The osmolar concentration or osmolar concentration. The unit used in hospitals when you give your patient a solution is the liter. You give him a liter of certain solution, half a liter, 2 liters. So let's make it the osmolar concentration related to the liter. The osmolarity is the number of osmoles per liter. This is what we use, the osmolarity. But they have another term, osmolality, is the number of osmoles per kilogram of water. One may say it's a little bit confusing. If you hear me in a good way, you will never forget it. You will remember what I am saying now. The osmolarity, it's the number of osmoles per liter. So the R with the R. The osmolarity is the number of osmoles per liter. And the osmolality is the number of osmoles per kilogram of water. Normally, 1 liter of water weighs 1 kilogram. So normally, the osmolarity equals the osmolality. This is just for discussion, not in real life. In hospitals, we use the osmolarity because to prepare a volume of 1 liter is very easy using a cylinder. But to prepare a kilogram weight of water, this is difficult to weigh 1 kilogram of water. But I tell you something, this what do we use in practical, but it's less accurate. You know why? Because the volume is affected by the room temperature. The volume of water expands in hot weather and shrinks in cold weather, while the weight is not affected by room temperature changes. But this is more accurate. However, because in hospitals at room temperature there is little variation in this room temperature, all hospitals all over the world are using the osmolarity because it's easier to prepare 1 liter volume than to prepare 1 kilogram weight of water. But you know it as a multiple choice fact. Osmolarity is the number of osmoles per liter, osmolality per kg. Now we come to the problems. Imagine I brought 1 liter water. Then I put in this 1 liter water 1 mole of glucose. In case of glucose, 1 mole gives rise to 1 osmole. There is no ionization. So the osmolarity of this solution is 1 osmole per liter. The second step, I try to make it a little bit difficult every time. I brought 1 liter water and I put in this 1 liter 1 mole of sodium chloride. What is the osmolar concentration of this solution? Smart student said a mole of sodium chloride will give 2 osmoles because each particle, each molecule of sodium chloride will be ionized into sodium and chloride. So the number of particles will be doubled. So the osmolar concentration of this solution will be 2. The third one, I brought 1 liter water and I put 1 mole of calcium chloride. 1 mole of calcium chloride will give rise to 3 osmoles. So the osmolar concentration of this solution is 3 osmoles. I tried to make it a little bit more difficult. In the same 1 liter, I put 3 things. 1 mole of glucose and 1 mole of sodium chloride and 1 mole of calcium chloride. What is the osmolar concentration of this solution? 1 mole of glucose gives rise to 1. Sodium chloride 2. Calcium chloride 3. So the sum will be 6 osmoles per liter. And you know what? In osmolarity, you don't have to say per liter. When we ask you what is the osmolarity of this solution, you can say 6 osmoles. You don't have to say per liter. We know that the calculation is always in osmolarity is per liter. The most difficult one. We have 1 mole of glucose, 1 mole of sodium chloride, 1 mole of calcium chloride in half a liter. What is the osmolar concentration of this solution? Now we have 1 mole of glucose gives rise to 1. Sodium chloride 2, calcium chloride 3. The sum will be 6. 6 in half a liter. The osmolarity is not calculated in half a liter. We have 6 in half a liter. So if we have 1 liter, 2 halves, how many osmoles will have? Double the 6 will be 12. I think very easy. Because the calculation always per liter. Now we have half a liter. We have 6 in half a liter. If we have 1 liter of this solution, the half contains 6, 2 halves will contain 12. So always I give it to you in a very simple way. If the volume of solution in the problem was half multiplied by 2 was quarter, that's 250 milliliters multiplied by 4. You know why? Because the liter is 4 quarters. If it's per 100 milliliter, multiply by 10 because the liter is 10 times the 100. So it's easier. I gave you all possibilities. In half a liter, multiply by 2. In 250, multiply by 4. In 200 milliliters, multiply by 5 because the liter 5 times the 200. If it's in 100, multiply by 10. I think it's very easy. We come to the problem. Before we come to the problem, we come to the 7th point. What is the relation between the osmosis and the osmotic pressure? We already said. Suppose that we put 1 millimole of glucose. Before I say that, I have to explain to you. We said in the beginning of the lecture that the osmolality of the body fluids 290 milliosmoles. Now you know how they did calculate it. How they did calculate this. They have 1 liter of plasma and they calculate this 1 liter of our body plasma contains glucose that much, sodium chloride that much, calcium chloride that much, and all other solutes in the 1 liter of plasma. And they calculate every solute and they have the sum by the end. If we have 1 liter of plasma and then we calculate it contains that much of glucose, that much of sodium chloride, that much of calcium chloride, potassium chloride, everything. By the end, the 1 liter will have 290, open your ear, milliosmol. This is the concentration of the body fluid. Intra, extra, or plasma. And you know all are the same. You know why? Because the cell membrane and capillary membrane are freely permeable to water. So water gets out and gets in until all are the same. The intracellular equals the extracellular equals the plasma equals 290 millismoles. Now you know what's meant by the osmolality of plasma is 290 osmoles. They calculate in 1 liter all the substances present, as we did already with 3 of them. Now we have a very important point. If we have here 1 mole of glucose in 1 liter, imagine this is 1 liter containing just 1 millimole of glucose. The glucose is non-ionizable solute. So 1 millimole in 1 liter will give rise to 1 milliosmol. The osmolality of this 1 liter will be 1 milliosmol. And they did this experiment. And they saw how much pressure they need to stop the osmotic force caused by 1 milliosmol. It's just finding, and you have to remember that number. They found that they have to press by a pressure equals 19.3 millimeter mercury to stop the osmotic force caused by 1 millismol. The osmotic force caused by 1 millismol, to stop it, you need osmotic pressure which equals 19.3 millimeter mercury. I ask you a question. If we have 1 liter of plasma over here, think how much pressure we need to stop this osmosis. If we have 1 liter of plasma, then we will not have 1 only. We'll have 290 millismoles. So we need pressure 290 times 19.3. 19.3 multiplied by 290. It will be around 500 millimeters mercury. Now you know what's meant by the osmolality of plasma equals 290 millismoles per liter, or 5,000 millimeter mercury. Now we come to the problem. If you have a problem in the multiple choice, follow these 4 steps. You will never miss the problem. Answer. First step. If he asked you to calculate the osmolar concentration, if you have been asked about the osmolar concentration, the first step, you know that the osmolar concentration is always calculated per liter. So the first step, calculate the amount per liter. If the amount given to you was in half a liter, so multiply by 2. In quarter of a liter, multiply by 4. 200 milliliters multiply by 5. 100 milliliters multiply by 10. Now you get the amount per liter. You know that every molecular weight, every mole gives 1 mole if the substance is not ionizable. So if the substance is not ionizable, divide this amount per liter by the molecular weight of the substance to know how many molecular weight we have. If the amount, say, I give you an example, 360 milligrams of glucose. Then you divide by the molecular weight for glucose, which is 180. It will be shown in the problem. Don't worry. They will give you the molecular weight. You don't have to remember the molecular weight. In case of glucose, if you calculate the amount per liter, it was 360. You know that every molecular weight, which is 180, which is shown, will give you 1 osmol. Then 350 will give you how many osmol? 360 divided by 180 will give you 2 osmoles. This is if the substance is non-ionizable. But if the substance is ionizable, it will give you more. In case of sodium chloride, the particle, the molecule, gives 2 ions. So you multiply by the number of ions per molecule in case of sodium chloride by 2. In case of calcium chloride, every molecule of calcium chloride gives rise to 3 ions, so multiply by 3. Now you get the osmolar concentration. So to get the osmolar concentration, you have 3 steps. First, calculate the amount per liter, because the osmolar concentration is calculated per liter. Second, divide this amount by the molecular weight to get the number of osmoles, provided the substance is not ionizable. If it's ionizable, it gives you more related to the number of ions per molecule, multiply by the number of ions per molecule. Sometimes they ask you to give them not the osmolar concentration but the osmotic pressure. You know the relation that every milliosmol needs pressure. This you have to remember, which needs a pressure which equals 19.3 millimeter mercury to stop the osmosis. So multiply the osmolar concentration. If the osmolar concentration was 100 milliosmoles, multiply 100 times 19.3 to have the osmotic pressure. This is the osmosis, the 7 points of osmosis. 1 point is left for osmosis. I said it already, and I will repeat it again. Is that? Yeah. Remember, water inside your body is transported from one compartment to the second to the third by the process of osmosis. Water transport in the body occurs only by osmosis. The cell membrane and the capillary membrane both are freely permeable to water. This means or will mean that the intracellular fluid will have the same osmolality of the extracellular fluid, the same osmolality of blood, which equals 290 milliosmoles. You don't have to say per liter. You can say it without per liter. We know it's always per liter, 290 milliosmoles or 290 milliosmoles per liter, which equals, if you multiply it by 19.3, around 5,000 millimeter mercury, which equals 7 atmospheric pressure. 500 millimeter mercury equals around 7 atmospheric pressure. You have to remember these numbers for multiple choice questions. These are the 7 facts about the osmosis. The definition, how to measure the osmotic pressure by osmotic pressure, which depends on the number, what's mole, what's osmol. Number 5, what's the difference between osmolarity and osmolality. Number 6, and how to calculate the osmolarity or the osmotic pressure. And the 7th point is that transport of water across in the body occurs only by osmosis. Now I would like you to know one thing. Sometimes the ions move, and in turn the electricity moves from one cell to the second adjacent cell, to the nearby cell, or between the intercellular fluid and the intercellular fluid via a gap junction. This gap junction is called, is a protein unit, is called connexon because it connects two cells. It's hexagonal in arrangement. That is, it has 6 pieces, hexagonal arrangement. It allows the ions and in turn the electrical activity to be conducted from one cell to the nearby adjacent cell without being lost in the extracellular fluid. From the intracellular fluid of this cell to the intracellular fluid of that cell without being lost in the extracellular fluid. This connection is called gap junction or connexon. It is a hexagonal arrangement. It's protein. I would like to tell you something. Everything in your cell or in your cell membrane, in your cell or cell membrane, which is active, is protein. Protein is the active part in our body. Receptors are proteins. Enzymes are proteins. Channels are proteins. Carriers are protein. Again, receptors, enzymes, channels, carriers, all of them are protein. The more the cell or the cell membrane is active, the more its protein content. This is a correct multiple choice question fact. The active part in our body is protein, not lipid, not carbohydrate. The more the protein content, the more the activity for the cell or for the cell membrane. Very important for multiple choice. Now we come to 2 small parts. That's the active transport and the vesicular transport. Now we come to the active transport. We'll discuss 3 points in active transport. The active transport. 3 points. The active transport, compared with diffusion, by definition, it's diagrammatically the opposite. It is transport against gradient. Against, not with gradient, it's against gradient. And here I drew a person climbing up the stairs. This definitely will need energy. We need ATP. So all the second point, you can anticipate it from the first. Being the active transport, as the name even indicates, is transport against gradient, so it needs energy, it needs adenosine triphosphate. But for a more smarter student, and it needs as well a carrier protein with ATPase activity to break down the ATP and liberate the energy. So we do not need only energy, you have to say we need adenosine triphosphate and a carrier protein with ATPase activity. Now we come with the types of active transport. We can divide it in many ways. We have primary active transport and secondary active transport. I always give this very simple example. The train and the train car or wagon going uphill, so we need energy to go uphill, but the energy is paid only in the train, in the engine of the train here, and the train is connected to the car or the wagon and pulls it up. It needs energy. It will not go by itself, but the energy is not paid directly for in the car. It's paid in the train. So the train is called the primary active, and the car or the wagon is called the secondary active. Definitely, if we stop the primary active transport, the secondary active transport will stop in turn. This is the simplest way to understand what is the difference between the primary and secondary active. You pay energy to get the train and its car up, but the energy is paid only on the train. This is the primary active transport. But the car or the wagon is the secondary active. But you have to connect the train with its car. What connects inside the body the primary active with the secondary active is the carrier protein, which has ATPase activity, which destroys the ATP to liberate energy for the train. The famous example of this for the train is the sodium. The sodium is the primary active, and it pulls with it the glucose. This is the secondary active transport. We call it secondary active transport or symport. Sym means connected because it's connected to the primary active. We call it co-transport. Co means with the primary active. This is the simplest way to understand what's meant by primary active and secondary active. The story of the train and its wagon. The train is the primary active, where the energy is utilized. The wagon needs energy. It will not go uphill by itself, but it has to be attached to the train. But the energy is not paid directly in the wagon, it's paid in the train. This is the secondary active. It's called symport. That's sym means connected. It's called co-transport because it's with the train. The best example is the sodium, and here we have the glucose. We can divide the active transport according to the type of carrier protein into three types. If the carrier protein carries only one substance in one direction, we call it uniport. Uni means one substance in one direction, and the example is calcium pump. If the carrier protein carries two substances in one direction, like our example, sodium, the primary active, and the glucose, the secondary active, we call it symport. If the carrier protein carries two substances in two different directions, sodium to the outside of the cell and potassium to the inside of the cell, we call it anti. Anti means opposite, antiport. So the active transport is divided in two ways: primary active or secondary active. According to the carrier protein, it's divided into three parts: uniport, symport, antiport. I say it in a very easy way. The uniport, one substance in one direction, uni. Example, calcium pump. Symport, two substances in one direction, like the sodium and the glucose. The antiport, two substances in two different directions, like the sodium and potassium. We come to the most famous type of active transport in our body. That's the sodium-potassium pump. Because this sodium-potassium pump occurs, open your ears, in every body cells. In every body cells, we have to keep the sodium outside the cell and the potassium inside the cell, as we already said in the introduction. The ions inside the cell start with the letter P, potassium positive. It's not our subject now, protein and phosphate negative. Outside the cell, sodium and chloride and bicarbonate. You can imagine that some sodium ions leak to the inside of the cells, and the same, some potassium ions leak to the outside of the cell. We have to bring this sodium outside in its correct location, and this potassium ion to the inside of the cell in its correct location. This is the function of the sodium-potassium pump, which is an antiport, because it carries two substances, sodium and potassium, in two different directions. The sodium to the outside, the normal location, the potassium to the inside, the normal location. Here we need two things, as we already said. We need ATP and carrier protein with ATPase activity. For any active transport, we need two things: carrier protein and adenosine triphosphate and carrier protein with ATPase activity. Now we start with the carrier protein. This carrier protein is composed of two units, alpha unit and beta unit. The alpha unit has got two characters. It has 3 binding sites for sodium inside and 2 binding sites for potassium outside. The beta subunit has ATPase activity. So this carrier protein has 3 characters. The alpha has 2 and the beta has 1. The alpha has 3 binding sites for sodium inside and 2 binding sites for potassium outside. The beta subunit has ATPase activity. I always say it in a musical way. When all sites of the alpha unit are saturated, the 5 sites in alpha subunits are saturated, the 3 for sodium and 2 for potassium, the ATPase is activated and energy is liberated. Again, it's musical. When all 5 sites of the alpha subunit are saturated, the ATPase of the beta subunit is activated and energy is liberated. This energy will cause configurational change in the carrier protein to get the sodium outside in its normal or correct location and the potassium to the inside in its normal or correct location. This sodium-potassium pump serves 2 functions inside the body. Number 1, it maintains sodium outside and potassium ions inside. Number 2, notice 3 sodium and 2 potassium. So the outer surface is more positively charged. This is what do we call the resting membrane potential or the polarized state. Resting membrane potential. In most of body cells, the outer surface is more positively charged in relation to the inner because the sodium-potassium pump pumps 3 sodium and 2 potassium. So the outer surface is more positively charged in relation to the inner. This is the resting membrane potential or polarized state. And this is why we call this pump electrogenic pump, as it generates electricity, it creates electricity with the outer side more positive. In multiple choice question, you have to know the sodium-potassium pump is an electrogenic pump, correct statement. Is an anti-port pump, correct statement. Anti-port because in 2 different directions. Electrogenic because it creates electricity. The third function for sodium-potassium pump, it controls the cell volume. If we poison the ATPase and stop the sodium-potassium pump, more sodium will collect inside than potassium. 3 sodium and 2 potassium. So the osmolality inside the cell will increase. This will drag water to inside of the cell. The cell will swollen and eventually rupture. What keeps the cell volume is the sodium-potassium pump. This is the most important pump in our body. It maintains sodium outside and potassium ions inside the cells. It's one of the 2 causes for resting membrane potential or polarized state. It controls the cell volume. We come to the last page. The third type of transmission is the vesicular transport. We talk about the vesicular transport in 3 points. I want to be very organized. By definition, it is the transport of macromolecules, very, very large molecules, cannot pass through the protein channels, and definitely lipid insoluble, across the cell membrane bound vesicles, across vesicles in the cell membrane. This is why it's called vesicular transport. That's transport with vesicle, within a vesicle. By definition, it is the transport of macromolecules across cell membrane bound vesicles. Again, it's an active process, that is, it needs adenosine triphosphate. I remind you for the 4th time, the only type of transport which does not need energy is diffusion by all its 3 subtypes: simple diffusion, facilitated diffusion, and osmosis. The simple diffusion and facilitated diffusion for solutes, the osmosis for water. The simple and facilitated diffusion or carrier-mediated diffusion for solutes, osmosis for water. Diffusion by all three types is passive, while active transport and vesicular transport are active. Now we come to the third point. Sorry, we will discuss 4 points. Mechanism and types. Now the macromolecule will come in contact with pit, not any part of the cell. The pit represents the mouth of the cells. The substance has to come in contact with the mouth of the cell. What do we call the pit? If this pit has the receptor, we call it coated pit. For the first step, as if you are catching an apple and approaching it to your mouth. This is the first step, comes in contact or attach it to the receptors in case of coated pit. The second step, the substance, the molecules of the substance are invaginated. We'll get into an invagination of the cell membrane will occur to get the substance inside this invagination. This occurs by means of pseudopodia. The third step, the membrane will break and fuse. The membrane will break and fuse to form a vesicle. Now the substance is inside this vesicle. The third and the last step is the digestive system of the cell, which is the lysosomes, one of the cell organelles, attracted to the vesicle and pours its enzyme, the lytic enzymes, inside the vesicles to digest the content of the vesicles. The good part of the digestion, the beneficial part, will diffuse to the plasma of the cell, which is the blood of the cell. The bad part will get rid of the bad part by a process diagrammatically the reverse to the endocytosis. We call it exocytosis or cell vomiting. The cell will vomit what is not needed by the cell by the process known as exocytosis. This is the difference between the cells and the human beings. The cells have no anus, so the cells have to vomit the things not needed by the process opposite to the endocytosis. Endo means inside, by the process called exocytosis, that is outside the cells. Here inside the cell, here outside the cell. So the mechanism and types, we have 4 steps and 2 types: endocytosis or exocytosis. In every cell, this is an important multiple choice fact, that to keep the area of the membrane, the endocytosis is balanced with exocytosis. The endocytosis equals the exocytosis. We can consider it continuation of the third point or the second point. The endocytosis and exocytosis can be divided further into other types. The endocytosis is divided into pinocytosis or cell drinking, or phagocytosis or cell eating. Let's see what is the difference between pinocytosis and phagocytosis. Endocytosis can be pinocytosis or cell drinking, or phagocytosis, which is cell eating. Actually, in both cases, the cells eat. But if the molecules eaten by the cells are invisible, what do you see? You see not visible. You cannot see the molecules under the microscope. You see some water entering the cells. You are not aware that inside this water there are protein molecules, but you see the water only. So it's called cell drinking or pinocytosis, despite the fact that the cell eats, but eats invisible things under the microscope. The cell eats substance in solution, not seen by the microscope. So you see only water, so it's called cell drinking. But in case of phagocytosis, which we call it cell eating, the cell eats large particles like bacteria or dead tissue. Here, bacteria and dead tissue are not in solution, not soluble in water. So you can see these bacteria or dead tissue under the microscope, seen by the microscope. So we call it phagocytosis. Let's compare between them in 4 points. Pinocytosis or cell drinking. Here, the cells eat macromolecules. The secret in the word molecules. Molecules not seen by the microscope. You cannot see protein molecule by microscope. So molecules are in solution, water soluble. So you see water only entering. Although the fact that water enters the cells together with the protein molecules, which you cannot see under the microscope because it is in solution, soluble in water. Every cell takes protein. So this pinocytosis occurs in all cells. This is the first point. While phagocytosis or cell eating, the cell eats large particles like bacteria and dead tissue. You can see bacteria and dead tissue under the microscope. They are not soluble, they are not in solution. The substance not in solution. So we call it phagocytosis. Not every cell in your body can phagocyte bacteria and dead tissue. Only phagocytic cells, like the white blood cells, the leukocytes, the macrophages, which are originally monocytes, or the microphages, which are the neutrophils. These are 4 points very important for multiple choice. The difference between pino and phagocytosis. Molecules, not seen, so in solution, all cells take molecules like proteins. Here, particles, large particle, the difference between molecule and particles, like bacteria and dead tissue, seen, not in solution, not soluble, only phagocytic cells. So types, we have endocytosis, to get the substance inside the cell, or exocytosis. Endocytosis already divided into pinocytosis and phagocytosis with the 4 differences. There is one multiple choice question fact. In exocytosis, the secretion of protein hormones from endocrinal glands, the endocrinal glands actually, the endocrinal gland cells get the hormones outside the cell, actually as if they vomit the protein hormone by the process of exocytosis. For all protein hormones are secreted from the endocrinal cells of their corresponding endocrinal glands by the process of exocytosis. Here we have another classification. If the pit has receptors, we call it receptor mediated. If the pit has no receptor, we call it constitutive vesicular transport. The receptor mediated definitely will be more specific, because the receptor site can unite only with the specific substances, as we explained with the carrier protein, and it will be more rapid. The receptor mediated compared to the constitutive vesicular transport, because of the presence of receptors, they are more specific, and they are the receptor mediated vesicular transport is more specific and more rapid compared to the constitutive one. This is the vesicular transport. By definition, it's related to the macro via vesicles. It's an active process, number 2. Now we come to the mechanism. As if a person eating something comes in contact with the pit, then invaginates, then go to a vesicle, then digested, and the remaining part we will get rid of by the process of exocytosis. And we come to the types. We have endocytosis and exocytosis. The endocytosis may be pino or phago. Molecules, pino, cell drinking, because not seen in solution, all cells can take protein molecules. Phagocytosis, large particle, not molecules. Seen under the microscope, the bacteria and dead tissue. Not in solution, not soluble, only phagocytic cells like white blood cells. And if the pit has receptors, it's rapid and specific, and we call it receptor mediated. If the pit has no receptor, we call it constitutive. Thank you, and we meet soon in another lecture. Thank you.